Okay, so I should start with how I experienced the AlphaFold revolution. And it started with a bit of anger. And the next stage was a bit of denial, you know, like it's one of these meetings that you're invited to share, or these journals that you will be the new editor. And it went on with bargaining because, you know, like as you're closing retirement as a crystallographer, I thought it's a bit too early. And the natural stage is depression because we all live on grants. And I felt as a structural biologist that, you know, the future was looking a bit bleak. And what followed was acceptance. And what better way to accept it but write a review article? So we wrote this article. We were invited actually by Yambo to write this article together with my colleague Tizia Sixman. And this is the point that I started thinking again about what AlphaFold is bringing. And I honestly realized that AlphaFold is great. Artificial intelligence algorithms for predicting protein structures are great opportunities, not only for experimental structural biology, but for structural biology in general. And I would like to give you an example before I go on in AlphaFill. I'll give you a couple of examples of how structure prediction affected our real-life projects in structural biology. And this is an example that we had determined a structure like a decade ago. It's about 160 residues. This was a part of a bigger protein, and it is blue domain right in the middle is the structure that you see on top. And we realized a few years later that actually also with that if we splice out this domain and we fuse the N-terminal and the C-terminal part of the structure, that form of the protein was enzymatically active because it's an enzyme and a DNA binding protein. And actually this was the only other truncation form of that protein that we were able to make. And at the time we did some small angle scattering measurements, and we showed that this small domain is quite flexible with respect to the main catalytic domain that is made by the N and the C terminus alone. So we were actually very happy to see the AlphaFold structure that has modeled this helical domain that you see on the right of the picture, connected with two links with respect to the main enzymatic domain. And what was also extremely impressive is that if you see the beta sheet in the middle of the big domain, the big blue domain, it is made 4 of the strands are from the N-terminal part, and the other 4 strands are from the C-terminal part. So that was not only an impressive prediction, as many, many predictions that you've seen also in the previous talks, but it was also like confirming our experimental work of more than a decade, and a few other things that I have not shown. What was even more remarkable is that when we looked at these PAE plots, and you can see actually what I just said at the PAE plot level, that the N and the C terminus form a sort of a rigid body that's moving together. In the middle, it's the other. And in the terminus to the left, where the big red arrow is pointing out, it was sort of telling us that these N-terminal residues are synergizing with a DNA binding domain. And we formed a very simple hypothesis that if these 50, 30 about N-terminal residues seem to have co-evolved and are in a relative similar position as the other part of the domain, could they also be involved in DNA binding? And indeed, this is we deleted these residues, and we showed through these assays that actually this N-terminal sequence is involved in DNA binding, something we have not suspected for nearly 2 decades. Another example of what we have done is that there's a protein that's called spindly that I've been working with my friends and colleagues here, Kops and Andrea Musacchio. You know, at some point we told the structure a few years ago of the first 100 residues, that was a coil, very clearly the diametic coil. But then we were looking at the SACS data because that's a 600 residue protein. Is it all of it a coil? It was not making sense. It was looking much more bulky, but pretty long. And of course, the AlphaFold database structure was useless for the monomer, but then AlphaFold multimer came in. We knew that it was a dimeric coil. We modeled it as a dimeric coil. And we got this beautiful structure that the coil, in some breakpoints, it folds upon itself. And this fitted beautifully with cross-linking mass spectrometry data that was indeed showing cross-links between distant regions of the coil that we could not have explained. Well, we were suspecting that the parts of the coil are falling into each other, but we were wondering if these are like trans or cis interactions, inter or intramolecular. And the AlphaFold model was amazing in clarifying this out and actually joining up with Andrea and putting together all this data with some beautiful mechanistic explanations of how this protein functions in events in early mitosis in the so-called corona expansion and contraction. Another project that actually, again, I found very impressive for how AlphaFold helped us is that we had the structure of these two repeat domains of tallin, 5 helix bundle, and a 4 helix bundle, and there is another helix that binds to the 4 helices. Very interesting puzzle for molecular replacement, by the way. And it was a bit strange because when we, you know, this protein tallin has 13 repeats, R domains. And when we deleted the domain with which we knew there was the interaction, that did not abolish the interaction, it enhanced it. And we wondered, you know, like, how could this happen? So what we did there is that we got the sequence of each and every R domain, and we tried to predict through AlphaFold multimer if it would come with with a good PAE matrix again, suggesting an interaction. And this suggested that some of the R domains, there is no data that ecoevolution again in reality, that does not support the interaction. For some other repeats, it was supporting an interaction. And indeed, that gave us the idea that mainly there are 11 and there are 12 repeats are also likely binders. We confirmed that experimentally. And again, AlphaFold helped us to generate a hypothesis and refine actually the biological hypothesis and make our experimental life much, much easier. So while doing all these, I also came to my attention and other people's attentions, of course, that there are also things missing from protein structure prediction. And this, what you see to the left, is the AlphaFold model for hemoglobin. And it has no heme, and it's a monomer. And biologically, this does not make any sense because we all know that the more accurate description of hemoglobin is to the right, that you see in different colors the tetramer, and you see the heme, and you see a nice big fat iron in the middle. So that's what gave rise to this AlphaFill idea that there's missing cofactors, there's missing ligands, and we know so many crystal structures, of course, and NMR and cryo-EM structures as well, that we could use this knowledge for transplanting ligands, natural ligands at least, and cofactors and metal ions from the PDB models, from the experimental models to all these predictions. So we made some choices for what we would be transplanting. Just to make a long story short, We avoided things that we know that there are crystallines, like, like glycerol, PEG ions. And we ended up that we decided that about 95% of all occurrences of ligands of the PDB are interesting molecules that we could be transplanting. And an example is heme, another example is ATP, and so on. So we're using a list of about 2,700 compounds that we're transplanting towards all the AlphaFold models. And that's an embarrassingly simple algorithm, which it's putting bits and pieces together that we get an AlphaFold model that has no cofactors. We blast the sequence against PDB-redo. Why PDB-redo? Because we like it. It's pretty accurate, and we like it because we are maintaining and developing it. Then we get the hit list by E-score with some criteria of the size of the domain and so on. And we check now the PDB structures that are likely homologs of this AlphaFold model if they had compounds that are in our transplant list. And we do a bit of cleanup there, and we come up with a transplant list. Then we do a quick structural alignment using quaternions between the PDB and the AlphaFold model. We get the aligned models, we calculate some RMSDs for validation, and then we transplant the compound from the PDB to the AlphaFill model. And as a result of that, if you go to the AlphaFold database and you look for myoglobin, for example, you will see that the two histidines are perfectly in place. But if you go to the AlphaFill model, you will also see the heme, you will see the iron, and you will see a few oxygens and monoxide atoms that might actually be there. When we did that work, that was the time that there was only 1 million models to AlphaFold. And this resulted to about 600 of them. We could find at least one ligand that would fit into them, and about 12 million transplanted compounds. When AlphaFold got expanded to 240 million, we decided not to do it, quite honestly, because this could be quite a big waste of electricity, calculating things that would not be very likely for people to use, but we still give the opportunity of people to do it. A big issue there is validation, and how are we sure that we're doing a sensible job, of course. And for that, we chose transplanted compounds that were transplanted from sequence with 100% identity, which means that although we put the compound in an AlphaFold model, the exact same structure has been determined experimentally for that domain. And when we defined a ligand validation score that we local environment validation score, the LEV score, which is the RMSD for non-hydrogen atoms in the neighborhood of the ligand. So this is a score that we can only know when we know the correct answer, the experimental answer. So the question we ask is that, does this LEV score correlate with metrics that we can know, that we can compute without knowing the correct answer? And for that, we chose the local RMSD, the root mean square deviation of the superposition in a sphere around the ligand, and actually also a new score that we defined, the transplant class score, that basically is telling us if there are classes of the ligand within the local environment following the transplantation. And when we check these scores that we can calculate without knowing the answer against the LEV score, that is when we know with the answer, they show the good correlation. So we decided that these are the local RMSD and the TCS are good validation score for the success of this transplantation exercise. That's also the point that we have a couple of ways for making to everybody accessible these models and the success metrics, the validation metrics associated to them. And this is either through our own AlphaFill interface, but also in a very nice and very efficient collaboration with the macromolecular structures team at the EBI, we also have a 3D beacons interface in the EBI knowledge base, and you can also access the AlphaFill models together with AlphaFold models if you use the Uniprot, the knowledge base, the EBI knowledge base. And you see that in our another feature is that if you are particularly interested in a particular compound, we also give you access through a quick optimization through Yasara. And we also showed for a few answers that for a few examples that if we find ligands that have either a low or a pretty high TCS score, we can improve at least the TCS score, i.e. reducing the classes, and very likely getting a more optimal and realistic representation of the ligand. This is a very, very simple and rough energy minimization, but makes life a bit easier if you want to use this structure for generating some hypothesis. Also, I should advise for quite a lot of caution there. It's a handy tool. Another thing is that these days you can not only retrieve these models for 1 million structure if it's not there, you can simply upload your own predicted model, which could be also a multimer or a complex or anything, and you can run it through AlphaFill, and we will try to check every domain and fill up the possible ligands. You can also get AlphaFill on GitHub if you don't want to be using our CPU power and so on and so forth.
You have 2 more minutes.
Yes, I'll go on with examples in these 2 minutes. You see, like, these are successful predictions of atoms over here. This is a bimetallic enzymatic site. And so on. And I will use the last minute and a half to say that what happens if we don't have experimental structures available? And what we did here is that actually we trained an AI that we developed, and in which I think we have a very interesting abstraction of the protein model in terms of 9 different chemotypes that we came up with, in which we represent the protein structure as a 3-dimensional image. And to be exact, we represent protein structure as a 3-dimensional image in a grid. And as soon as you have something in a grid that resembles an image, that makes it quite easy to use out-of-the-box learning methods. And with a few tricks, like, we take a metal site. So you see in the middle of this cube, there is a metal site, and there is an environment around it. The gray is carbon, red is oxygen, and so on. And then within this environment, we use what we call the sampling cube to sample this environment, and the learning cube, which is a zoom in into this environment to learn how metal binding sites look from different views. And then we start learning, and within the learning epochs, we use the typical image manipulation tricks that we rotate the image around to create the rotational variability and so on. And we simply pass that to an out-of-the-box neural network used in many AI applications to predict the 5 most common metal sites on proteins. So we trained that on something like 11,000 proteins for 40,000 metal sites. And then we can get any protein, sample it with these learning cubes, come up with some probabilities for every point in this image to have a protein. We translate back to the protein coordinates. We use a few tricks of the probabilities to consolidate the answers, the probability answers that come out from the AI. And the bottom line is that now we can also predict the position of metals without directly looking at the homologous protein structures, but having learned, having taught an AI for how metal environments look like. And you see here some prediction that it comes up with. It's doing pretty well. It even gets bimetallic sites, which we're a bit surprised, but it's quite good on getting that. It also finds a few things that could be false positive, because here it predicted something when there was nothing there, but perhaps there is something really, a magnesium in this amine oxidase, or a copper over here is perhaps a false negative, perhaps not. And it finds, if Ed Snell is listening to me, he mailed me, in cases that you have nickel bound, we predict zinc, which is very likely the correct thing to be there. We also find zincs, and importantly, not only known PDB structures, we can recover that, but also in AlphaFold models, we can also retrieve metal sites like structural zinc sites. So quite a lot new with shiny irons in very well-known crystallographic structures. And of course, our dream, together with the dream of a few others, is that if we can use this AI that we develop for metal sites to predict the binding of larger molecules like fragments or ligands. Sorry for stepping a bit over time, but I will just show the names of the collaborators. Martin, who did a fantastic job implementing everything. Ida de Vries, who did all the validation, and her name is not there because I stole her acknowledgement slide and forgot to add it, as I now realize. Ren came up with AI algorithm, and all these projects have been co-supervised, as always, with Robbie Yosten. And I should also mention that the fact that CCP4 has supported and that is supporting Robbie has been fantastic, and it allowed us also to indirectly do things like that. Hope you visit the site, don't cross it. We got 3,000 requests this morning from a single person. Don't do it. But please go and check the website. Sorry for being a bit over time, and thank you for your attention. I hope I did not tire you in the last talk of the meeting. I wish I was there. I hope you all had a fantastic time. Thank you again.
Thank you.